We're very fortunate today to have Dr. Todd Druley, who is the CMO of Mission Bio. He's joining me in our Cota Access webinar. Mission Bio is one of our really exciting health tech portfolio companies, and I joined their board in May of last year. So Todd, I'm going to let you introduce yourself.
Thank you, Janice, and thank you to Cota Capital for having me as a guest today. It's a pleasure to be here. So I am a pediatric oncologist by training. I've trained at Washington University in St. Louis, where I was really intrigued by the work that they were doing in the Human Genome Project and developing genomics for patient care. And so I did that for about 15 years. I had my own genomics laboratory, and then had an opportunity to step in as the first chief medical officer for Archer DX back in 2019. And so that was an unexpected but fantastic opportunity. And as I've watched technology evolve, you can always think about, well, what would be the next plateau for patient care as we bring more and more technology to bear, especially in this era of precision medicine, which I know we're going to talk about. So I joined Mission Bio about 1.5 years ago, thinking that single-cell multi-omics, particularly DNA and protein, are the future of cancer care. And so it's been really exciting and very challenging. So I'm happy to be in this role. Thank you.
So Todd, I'm going to just deviate just slightly a bit now, put you a little bit on the spot. You do have an MD and a PhD. So can you make a little bit comment about, why, how, both, and kind of what that brings to your abilities and your interests?
Sure, yeah. According to my parents, when I was about 4 years old, I wanted to be Batman. And then shortly thereafter, I decided to be a doctor. And I don't know what prompted the switch, but I stuck with it. And I always wanted to be an MD. As I got into college and, I did more research and I just loved it. And so I had the opportunity then to do a combined MD and PhD program at the University of Illinois. And that was in Chicago. So that was, again, an unexpected but fantastic opportunity for my own personal career development. And during that time, my cousin had a 3-year-old son who got AML, leukemia, and he was at Riley's Children's Hospital in Indianapolis. So I drove from Chicago to Indianapolis a few times to visit, and unfortunately passed away waiting for a bone marrow transplant. And so while I was doing genomic research and protein biology research on cancer in general, and drug resistance in cancer, I saw the personal effects, from a family member for pediatric cancer. And that's what really drove me to get into pediatric oncology. And I can say having the PhD gives you a very, I think, it gives you a foundation in how to vet, clinical trials, how to vet technology, and, really build, the data sets that are necessary to be, I think, convincing in terms of, what's next, what's working, what's not working. You can sort of cut through a lot of the hype and get straight to the heart of a matter in a way that sometimes you can't otherwise, in my opinion. That's
good. Well, I remember when I was doing my PhD in pathology at USC, and I came into contact with a number of physician researchers. And I have to say, there's some of the best scientists I've seen out there. And what I really like about it is that it's really they apply the science and they really touch the patient. So I've always thought that, even back then and today, that, if you're going to be doing research and you're going to be building companies in this space, you got to have somebody like you on the team. So I know Mission Bio is very fortunate to have you, and I've always enjoyed interacting with you. And I think that story you told, thank you for sharing that. I think that also really speaks to kind of, what's the North Star for what you do. And I think that's very, very important. So we hear a lot about precision medicine. I throw that term around a lot myself. But, getting down to kind of the meat of it, what exactly does precision medicine mean to you?
Yeah, I think that's an intriguing question. When I was still practicing, I would talk to parents of children who had cancer. We were a very advanced cancer center. And so I would tell them, one of the advantages of being here is that we're going to do our very best to bring precision medicine to your child's care. How do you feel about that? And almost exclusively the answer was, that's great. That's why we're here. We want that. And they see it as a very positive thing. And then I would flip the question a little bit and say, well, what if I, challenge you this way and say, I'm going to order a bunch of tests for your child. I'm going to review these tests myself and with some of my colleagues, and we might design a treatment plan for your child that we've never offered to any other child because we think it's precise for your child. And how do you feel about that? And then the answers were very, very different. It was, I'm not quite so sure that's exactly what I'm comfortable with. And so precision medicine means different things to different people. And for myself personally, it means bringing the right therapy to the right constellation of disease features, whatever that may be. And so I've always been very, very intrigued by the marriage of technology bringing treatments into the right patient context. 5 or 10 years ago, there weren't that many precision therapies to choose from. So the technologies that you used to match a patient with a treatment didn't have to be all that sophisticated, quite frankly. But now we've got roughly 75 approved drugs. We've got another maybe 50 to 75 approved cell and gene therapies for all different kinds of conditions. And so we need better technology to bring the right therapy to the right patient and the right disease characteristics. And that's only going to continue to grow as far as I can tell.
No, I think that's an important kind of point to make, right? Because certainly as a venture capitalist, that term is well understood by venture capitalists, at least in terms of raising money, right? Precision medicine is, the kind of the important way to go, right? And certainly even, when I started out myself in graduate school making monoclonal antibodies, that was kind of early days of precision medicine. So, when you come from that kind of background, you're like, okay, just that's the word, right? So I think, again, just hearkening back to what I was saying about being a physician researcher like yourself who really talks to patients, I think you brought up a really good point. Is like, we really need to make sure that it is the patient that understands that we're trying to bring the best care to them possible. And it's really important as a physician that you're translating that. So that's a really important point to make. So that does kind of bring us to, so tell me more about tell the group more what Mission Bio does. First of all, why Mission Bio? That you decided to come to, and then also, what do they do? Try and explain it so that people can understand it, and then how does it apply to the area of precision medicine?
Sure. Yeah. So, the whole genomics era started in, probably around 2000 to 20, 25 years ago. And, this is, for lay audiences, I like to say this is when we went from analog television where you could see a little bit of fuzz and snow. We went to digital TV and everything was crystal clear. And that's the difference in the amount of information we're gaining in genomics compared to genetics. We're able to survey huge areas of the genome and the transcriptome and the methylome, and that was only made possible by supercomputing capabilities. So the computing capability to integrate that much data in a time-sensitive fashion made all of this possible. But then the next question is, well, how do I interpret this, and what does this mean for patient care? And so all of that sequencing that we've been doing for the last 20 years is what we call bulk sequencing. So if I take a sample, whether it's from a tissue or an organ or bone marrow or blood, and I want the DNA, I'm going to burst open all the cells in that sample and I'm going to take the DNA out, but all of those cells get lumped together and I get one amalgamated measurement of every cell in that sample. And that's bulk sequencing. And, you can do sort of low pass or non-sensitive sequencing. You can do sensitive sequencing, but at either way, it's bulk sequencing. And I can't with real definition tell you if this genetic change is actually in the same cell as a different genetic change. And then as we do more measurements, we may look beyond DNA and add in RNA or proteins. And I cannot tell you if that single cell has all of these features. I'm making an educated assumption across this bulk measurement. And so again, our need for more and more precision, high resolution technologies to match patient to drug is growing. And we're finding more often that lack of resolution is detrimental to patient care. It leads us to false negative results or false positive results, depending on the clinical context. So I've moved out of that sequencing and moved to Mission Bio because it's the only company that currently exists that is measuring DNA and cell surface proteins from the same cell at the same time. And so if we look at the history of how genetic tests have been regulated, there's never been the FDA or any other countries or a regulatory body that's approved RNA as a standard diagnostic measurement. And that's generally because RNA is pretty fragile. And so, it's going to be different from person to person, cell to cell, in different temperatures and different ways you manipulate it or freeze it or thaw it. And so it's very, very hard to take RNA and get the exact same result in place A, place B, and place C. DNA is much more stable and proteins are much more stable. And so those historically have been the two modalities, the analytes, we like to call them, that have been approved by regulatory agencies for patient care. And so, I see the future, if single cell multi-omics is going to be the future of patient care, and I believe that, and more people believe that, then it's going to be DNA and protein. And Mission Bio right now is the only company that can offer that kind of technology.
That's excellent. Certainly why we are really excited about the company as well. So I'm going to put you a little bit on the spot here. You do such a great job of explaining, some in more simple terms, understanding. So you went very high level, and then you kind of zoomed down to kind of a submolecular level and talked about DNA and RNA and omics and things like that. Can you kind of go up another level and kind of say, well, how does that kind of tie together in terms of just cancer? Right. Again, kind of putting you on the spot here. Why do all those pieces, translate to what, how cancer even evolves or grows or, changes over time? Can you just, talk to that a little bit?
Sure. Yeah. I'd love to talk to that. So, cancer is not one disease. It's hundreds of diseases. It may be thousands of diseases. So, we have historically thought of it in terms of, say blood cancer, but blood cancer is leukemias, can be lymphomas, can be myelomas, and myeloid, lymphoid, so we can go to all different cell types. And then we can have subtypes of those, and depending on their genetic architecture or other features of that disease. And we're doing this for every type of cancer: brain tumors, lung cancers, liver cancers, kidney cancers, skin cancers, breast cancers. There's subtypes, and then there's what we call molecular subtypes of every subtype. And so, the number of potential diseases that we're trying to treat is growing and growing and growing. Pharmaceutical companies are now developing precision therapies that are treating just that, particular subsection of that particular cancer. So, they're looking for technologies that will sort of boil down and say, is this right? And cancer is driven by mutations. And what is a mutation? It's a change. So cancer is always changing. So on the day that a patient is diagnosed with cancer, that cancer is going to change once we start treatment. And it's going to change differently depending on the treatment that we bring to bear. And it's going to look different depending on the technology that we use to monitor it. And so what I like about the single cell space is that we can watch that cancer change in a more comprehensive fashion in terms of the mutations that each cell has, they're not all the same. There's going to be 1% of cells that may have a resistance mutation. So if I kill 99% of the disease, that looks great on day one. 6 months later, that disease has come back and it's even more resistant now. Boy, I wish I'd known that on day one. I would have done something in addition to what I did to kill that extra small percentage of resistant disease. So these are the strategies that I think we need to employ. A lot of this is generating data, and that's where we're working with people all over the world to try to generate the data to prove these points. And we haven't proven these points yet, but based on how technologies come through cancer diagnostics historically, we think that we are in a great position to make these arguments going forward.
No, I like how you framed that, because really it does bring together kind of the intersection, right? Because you mentioned pharmaceutical companies that are developing very precision drugs in which they need this information. They need to work with someone like with Mission Bio, but really it does translate to helping the physician decide, how to treat the patient even over a given period of time. So really, Mission Bio is sitting kind of at the crux, at the intersection of that sort of knowledge. The other thing you brought up is data, right? And so, we've just been inundated with data. I think RBC, the bank, noted that 30% of the world's data is healthcare data. And I just recently got back from a health conference in Las Vegas, in which the corporate vice president of the Azure group at Microsoft said that a hospital can generate 50 petabytes of data per year, right? So there's so much data out there. And like you said in the very, very beginning, it's like, you have to interpret this information. So you, as you're sitting there, certainly within Mission Bio, and you're seeing the platform generate a lot of this data, and you're the one that's trying to figure out, okay, well, how do we interpret this data? How do we manage that data? I mean, I think maybe when you did your MD PhD, you didn't anticipate you were going to also be a data scientist. How do you think about that, and what do you see within Mission Bio in terms of how that's, what's Mission Bio doing in this, since you are at the intersection of generating all this really useful information to make really key decisions?
Yeah, that's a lot of complex issues there. So let me back up to physicians and how we want to help physicians. As I alluded to earlier, physicians now have more and more options for patient care. And so historically, when we think about disease monitoring, we think about high sensitivity. That means I can detect with high accuracy a very small amount of disease. And that will spur the physician to do something, and the patient will have a better outcome because something was initiated sooner than if we had used a less sensitive technology. Now, in some respects, that's harkens back to 70s, where we improved surgical methods with radiology, MRIs, CT scans, and so we would see an abnormal clump of something, and the surgeon would take it out, and they would say, "We caught it early before it spread," and so this was a good thing to catch it early. But now, we're finding disease that doesn't show up on any scan. It's molecular disease, microscopic disease that you can't see. And so physicians are asking, well, now you've told me my patient has a problem. What the heck do I do for this person? I don't know what to do. And in some cases, I don't want to add a treatment that's going to be very expensive or toxic or have other problems. And so I think that is where we bring something that doesn't exist more broadly in the marketplace today. We can say there is disease present, and here are the targets on that disease, and let the physician choose from the possible options there. Now, for us to do that gets to the point of data. We have to have an abundance of data to say that those decisions are safe to make and are driven by good data sets going into it. So, we have to build our own bioinformatics and computational resources to, I can tell you from the little data that I've seen that when you try to merge sequencing, I mentioned bulk sequencing, and I think, flow cytometry is another, where the cells move along and you're using a laser to measure the proteins on the cell surface. We're essentially coupling those two things but then giving single cell resolution to that. And when you're measuring all of these things in a new way, the numbers are slightly different than what we've historically become accustomed to. It doesn't mean they're wrong. It just, we're looking at this through a new prism. And so we have to build software that is setting new thresholds for when should we be concerned, when shouldn't we be concerned, and ultimately being able to provide a report to a physician that gives them clinically validated data that has good clinical context backing it up. And that's a long-term process. That's a big undertaking, but we're well on our way in some blood cancers, particularly AML and myeloma. We expect additional blood cancers to be added in the coming months to years. And so I'm very optimistic. The pharmaceutical companies, are using the same amount of money to generate the drugs, which we always know are huge amounts of money, but they're aiming that drug at a smaller and smaller subsection of patients. So it takes longer to find the right patients. And if you're not using the right technology to figure out who goes into your clinical trial and who shouldn't go into your clinical trial, having 5 or 10% of these patients inappropriately added or left out can skew the results of the entire study. So we're finding pharmaceutical companies are very enthusiastic about more comprehensive technologies to identify precisely the right patients to fit into these precision therapy clinical trials.
Yeah, no, having come from the pharma industry myself, I know how much money and time goes into these clinical trials. So the more data and information you have in terms of the patient stratification for the trials, the better. So I do want to give you a special prize for being able to go through all of that and not ever mentioning the word AI. So, but as we all know, you cannot get on the internet now or swing a dead cat, as I say, and have someone mention, oh, AI, and what it's going to be bringing to certainly the healthcare industry. So I am going to ask you, first of all, what do you see in the terms of, what does AI mean to you when people say they want to apply it in this situation? And, where do you see its utility and where do you see it? Maybe it's just kind of, not here yet and maybe just creating noise. So now you have to talk about it.
Okay, well, it's clearly it's early days for artificial intelligence. This would be when, we just generate the data at the laboratory and we feed it into some system that's going to aggregate who knows what from who knows where and spit out an answer. And, I think a lot of people think this will just automatically be the right answer. We've had a few publicly available artificial intelligence platforms that are, and if you were to search for mistakes that these platforms make, that some of that stuff is actually quite creepy to look at. That comes up with some pretty bizarre random stuff, images and interpretations and text and things that clearly are not, we know well enough to know that is not accurate and that's not reality. So if I was going to put, patient care decisions into the hands of an artificial intelligence, I would have to know that it is drawing its conclusions from highly validated, very accurate repositories of information, not just anything, anywhere, at any time. So that would be very frightening to me. And so, again, I'm not saying there's no usefulness there. It's just early days, and I'm not sure how we're going to vet the information going into it. It reminds me of when I was in medical school, everybody was excited about meta-analyses. We're going to take different clinical trials that were done in different ways by different people using different statistics, and we had some sort of way to smooth them out and get one result that would sort of rule them all. And it didn't work. And it was very clear that if you put garbage in, you would get garbage out. And so you cannot overcome poor source data. And so for artificial intelligence, if we're going to leave this up to a machine to choose patient care, it better darn well have very, very highly accurate source data. And we're nowhere near that yet.
Got it. So obviously, because we get pitched continuously by those who say that they have algorithms that can, predict all matter of things. So from that standpoint, obviously, first questions are, what's the data source, right? Many of times it's a public data source. Where two things, how critical are like proprietary data sources? And in the area of even, kind of cancer analysis, longitudinal data sources. I mean, can you give me like maybe three things that as a VC you counsel me to say, hey, look, ask these three questions, before proceeding further with this company?
Sure, yeah. I think the high quality data sets that are big enough to inform artificial intelligence are very few and far between. So you run into the potential that, what's unique about their algorithm that will generate a different result than the other 100 players in the market if they're all using the same source data. So to me, that's the first thing. But is that conclusion even accurate? Why would this conclusion be more accurate than the conclusions drawn from the same source data using a different algorithm? And so if you're using a proprietary data set that's been generated by a private company, those could be enormous in size, but where did their data come from? Is it from highly controlled, highly vetted, very consistently generated data, or is it just sort of drawing things from all corners of the globe? And again, the way things are done, we know this, right? I mean, this is the same reason we don't use RNA for broad scale approved clinical diagnostics, because you do it the same way at different places and you get a different result, and that's due to the inherent nature of RNA, and data can be very, very similar to that. So those would be the things that I would be cautious of.
Yeah, no, those are very good points. So maybe what we'll do is, I wanted to thank you for joining us. I think you've certainly made a great case, and I think hopefully educated people more in terms of precision medicine and how it all fits together. Maybe what we'll do now is get to maybe some rapid fire questions, I think, but in the meantime, I need to actually put our disclaimer on in terms of the, here we go, let's see. There we go. So, we need to post this disclaimer to let people read that. But I do want to thank you, Todd. Before we get to your rapid fire questions, I think it's been such a pleasure to be on the Mission Bio board and work with you over the last year and a half. I think that the work you've done is amazing, and I know that you've been traveling the world. And while that sounds really glamorous, I know it's a lot of hard work. But I think it just really speaks to the fact that there's many key opinion leaders and hospitals and doctors and pharma companies that really do, see the value here, and you are, a major component in terms of really, bringing this technology and these interpretations to the bedside. So why don't we go ahead and we can move to some rapid fire questions. You ready?
Let her rip.
Okay. So let's see. What is your favorite movie that you're really kind of embarrassed to admit to?
Yeah, I would have to say Love Actually, the Christmas movie. It's super sappy. It's a holiday movie. I get drawn into those characters every time, and I can't turn it off, and it's really, really embarrassing.
No, well, hey, I applaud you on admitting that, and I love that movie too, and I'm glad you didn't say some of those Hallmark holiday shows.
No,
that would have been really embarrassing. This is a little bit of a throwback, but I think you can step up to it. Back in the day, we used to listen to albums and not just stream our music. If you were going to get plopped onto a desert island with no streaming capabilities, what 3 albums would you take with you?
Wow. So, I I would pick 3 albums based on their style, to have 3 different styles of music.
Very thoughtful.
I think sort of chill music. I just love the album A Rush of Blood to the Head by Coldplay. So there's so many good songs on there. The Scientist is almost probably my favorite song ever. I love that song. So that would definitely be one. An album that's just chill, it's going back a ways, but it's Air, Moon Safari. It's mostly instrumental, but it's just very chill music that, I like to just relax to. But then you got to have a hard driving rock and roll album. And for me, that's AC/DC Back in Black. I just want to turn it up, rip the volume knob off, and let it rip. So that would be the 3.
Good for you. Okay, so a couple chill and then something to get you going. Excellent. All good choices. And the last question is, if you could have beers with 4 people, living or dead, who would be the 4 people?
Yeah, well, so the first I would pick is Barbara McClintock. So
Ah,
she
okay.
discovered what was she called jumping genes, and these
Yeah.
were turned out to be transposons. My dad did agricultural research on corn for his entire career. That's how I learned genetics, was working in a cornfield and mixing inbreds and making new hybrids. And so her work in corn was something I heard and read about quite a bit when I was younger, and very intriguing to me. And of course, she won the Nobel Prize for those efforts. So that would be one person. I love the movie Oppenheimer this summer, and I'm reading American Prometheus right now. And so I think right now I'd like to sit down with Oppenheimer and pick his brain a little bit. That would be fun. Winston Churchill is a guy who, really rallied from almost impossible odds, or what seems to be impossible odds, to victory, in World War II. So I think that would be a person who I'd love to talk to. But then someone living who just seems super interesting and has had a dynamic life and career would be Dave Grohl, the lead singer of the Foo
Oh, yeah.
Fighters.
Yeah.
So he just seems like a top quality guy. He's amazingly talented. He's been everywhere and done all kinds of stuff. So I think he'd be a lot of fun to have a beer with.
Yeah. Oh, again, all good choices, and certainly tells us a lot about you as well. So again, I want to thank you. This has been great. I hope you've enjoyed it. I've enjoyed it, and I hope the people participating in the webinar enjoy it as well. And I look forward to the continuing success of Mission Bio and getting together again over that.
Well,
So thank
thank
you very
you for
much,
your
Todd.
support. Yeah, thank you for your support and Cota support. It's been a pleasure to be here. I have had a lot of fun.
Good, good, excellent. All right. Well, I'll see you later.
All right, bye-bye.